Next, we'll talk about SAR studies. So, SAR, it is structure activity relationship. So, what do you mean by SAR? It is structure activity relationship. So, the relationship between the structure of the drug and its pharmacological activity is called as SAR of a drug. So, which drug we are going to discuss here is sulfonamides. Sulfonamides, which are also termed as sulfanyl-amides. So, these are the category of the drugs which we are going to discuss their structure, activity, and their relationship. So, what is the structure of this category of the drugs, and what is their activity, and what is the relationship between them? So, pharmacological activity or biological activity of a drug depends upon the structural features of a drug. Means which structural feature is responsible for showing the activity. That study we call it as SAR study. So, which structural feature of a drug is responsible for total activity of a drug to be shown. That study we call it as SAR study. It is generally useful in early stages of drug development by medicinal chemists to identify more promising drugs and to improve the properties of lead compounds and to produce more compounds with similar structure which is showing the activity. Means more promising compounds with ideal properties can be developed. So, it is very useful and very important in early stages of drug development to study the structure of a drug which is responsible for showing its proper activity, right? That study should be done in the early stages of a drug development to get more promising compounds and with more improved properties, right? And to categorize those properties into one channel, that study is very important, right? Examples, the properties are solubility, pH value, pKa value, and all those things, lipid solubility, water solubility, and all those things can be categorized into proper channels if a structure activity relationships is study is done in the early stages of drug development. So, which category we have selected is sulfonamides or sulfanylammides, which indicates the structure have the sulfur drug, means the sulfur atom is present in the structure of a drug. So, which activity this category of drugs will be shown is antibacterial activity. So, these are considered as first antimicrobial agents which have broad spectrum of activity to show activity against bacterial infections in both human and animals. So, these are considered as first antimicrobial agents. First antimicrobial agents or drugs which have broad spectrum of activity against bacterial infections in both human and animals. The first identified drug is Prontosil. Prontosil means the antibacterial drug commercially available in the market which belongs to sulfonamide category is Prontosil. It is the first antimicrobial drug identified. So, it will be in the form of prodrug which will get metabolized after entering into the body into the drug sulfonamide. So, this Prontosil, which is in the prodrug form, which is commercially available in the market as an antibacterial agent, will get metabolized after entering into the body into the main drug, that is sulfonamide, right? So, the structure of this Prontosil in the form of hydrochloride form will get metabolized after entering into the body into the main drug sulfonamide, that is NH2. This is the structure of sulfonamide. So this is prontosil considered as the first antimicrobial drug identified, which will get metabolized after entering into the body. So let's discuss about the main drug, sulfonamide. So the sulfonamide has the basic structure like this, right? It has one benzene ring, 5 or 6 membered aromatic ring with two substituents which are para to each other. If these substituents are placed ortho or meta to each other, means if they are placed in 1,2 position or 1,3 position, antibacterial activity of the sulfonamide will get decreased. So these substituents should be always para to each other. They should be at opposite to each other. That is first, fourth position, right? So that is a first thing. It has 5 to 6 membered aromatic ring. It has one aromatic ring, and the substituents should be at 1,4 position or para position, para to each other. This is the second point. And the third point is no substitutions or replacements should be done on this aromatic ring. Remaining position should not be substituted or replaced. This ring should not be replaced or remaining position should not be substituted. If it is done, antibacterial activity of this drug will be decreased. So, no replacement or substitution should be done. The replacement of aromatic ring or substitution on the ring should not be done. If it is done, the activity of the drug will be decreased. That is other point. Next, we'll talk about this amino group. So, this free amino group can be substituted by any other substituents. Means this amino group can be substituted by thalilamide, acetylamide, and all those things, sulfamido groups. So this free amino group can be substituted by many groups because it is reconvertible. Once it is substituted, it can come back. So this amino group substitution can be reconverted. So free amino group can be substituted as it is reconvertible. Right? So next we'll talk about this sulfur group. So this sulfur group should be directly attached to the benzene ring. It should not be linked by any other atom. Means sulfur and benzene ring should not be linked by any other bridge atom. So that sulfur should be directly attached to the benzene ring. Then only the activity of the drug will be retained. If it is attached by any linker, the activity of the drug will be decreased. Means the less potent compounds will be produced. Right? The sulfur should be directly attached to the aromatic ring. So next is the substitution at these two hydrogens which is attached to the sulfur group. So the amino group which is attached to the sulfur group is considered as the substitution at first position. The amino group at first position can be substituted by substituents which imparts electrons to the sulfur atom. Means the substituents should be electron donating groups. These hydrogens of this amino group which is directly attached to the sulfur atom should be substituted by substituents which are electron donating in nature. If the substituents are electron withdrawing nature, the electron rich nature of sulfur atom will be reduced. So the activity of the drug will be reduced. So the substituents should impart electrons to the sulfur atom. So that substituents should be substituted by these hydrogen atoms, and two hydrogens cannot be substituted. Only one hydrogen can be substituted because one hydrogen is responsible for ionization of the drug. If the drug should be ionized, one hydrogen should be retained. So only one hydrogen can be substituted. That substituent should also be the electron donating group. If it is not, the activity of the drug will be decreased. The electron donating groups substituents will impart electron rich nature to the sulfur atom, which increases the activity of the drug. so that high potent drugs or compounds can be produced. If the substrate has electron withdrawing nature, the electron withdrawing nature will pull the electrons towards itself, so antibacterial activity will be decreased, so less potent compounds will be produced. So because of that, the substrate should be electron donating groups. And the two hydrogens cannot be substituted, only one hydrogen can be substituted, because one hydrogen should be responsible for ionization of the drug. So the drug will be ionized maximum at a pKa value 6.6 to 7. And lipid solubility of the drug also imparts the activity of the drug. As the lipid solubility increases, the activity of the drug will also get increased. So this is all about the structure of sulfonamide drug, which is responsible for its antibacterial activity.